Right, just respecting everyone's time, we've reached 10 o'clock in Vancouver, and I would like to welcome you all to this webinar, looking at the UK's approach to precision prescribing, transforming traditional models of care with pharmacogenetics and the applicability of that work to North America. My name's Martin Dawes. I'm a professor of family practice at University of British Columbia, but also the scientific director of Genexus Healthcare Systems. With me today is Professor Sir Munir Pirmohamed and Dr. Bernard Esquivel. Before I just start the slides, Professor Pirmohamed, could you just give us a little bit about your background, and then I'll ask the same question of Bernard.
Thank you very much, Martin. So I am a clinical pharmacologist by background, and I'm a physician working in the National Health Service in the United Kingdom. So I work at the Liverpool University Hospital Foundation Trust. I hold the David Weatherall Chair of Medicine at the University of Liverpool. In 2007, I was awarded the NHS Chair of Pharmacogenetics. I've had an interest over a long time into individual variability in drug responses, and hence my work in pharmacogenomics.
Thank you. And Bernard.
Thank you, Martin, and thank you for having me. It's a pleasure and an honor sharing this webinar with you. Yeah, my name is Bernard. My background, I'm a clinician by training, clinical immunologist and medical geneticist by training. And I've been focusing my career the last 13 years around precision medicine specifically, and particularly around precision prescribing and pharmacogenomics for over the last 7-8 years. I've been involved in different fronts, from the industry to the academy, different regulatory affairs, mostly focusing my career in the US and North America. Currently, I'm with Genexus as Chief Medical Officer.
Thank you. So moving on, I have a question for you, Munir, which is really, I think, relates to why we're all here, but why did you decide to focus professionally on pharmacogenetics?
So I guess that goes back to the time I was a medical student. The first clinical attachment I did, I was a second-year medical student, and a chap came in to the emergency room, had pinpoint pupils, shallow breathing, and admitting officer, clinician, made a diagnosis that this patient had opiate-induced respiratory depression and gave him naloxone. And the person who was given naloxone woke up like magic. And I thought, wow, that's something I want to get into. Because of the power of something like that. And then I sort of decided to became interested in pharmacology, clinical pharmacology, and pursued a career track in that. And when I was doing my PhD, I had to identify an area I was interested in. I was interested, as I trained and got more experience in clinical medicine, I could see that there were people who were who were getting better with the medicines they were given, but many people weren't, and many people were getting adverse effects. And the reason for that variability was not, clearly everybody thinks about clinical factors determining that variability, but I was interested in some of the genetic factors also determining variability. And as I said, I hold the David Weatherall Chair of Medicine, and David Weatherall was very famous for the genomics work he did, and I remember reading his book on genetics. I thought, well, that's an area that I think I can do some work in and try to relate that to the medicine's use, because medicine's use is very common. Medicines are prescribed by people in primary care right through to tertiary care, and it's prescribed by many different healthcare professionals, not just doctors, and therefore it is important to a wide group of people. And I see pharmacogenomics as an area where you can actually mainstream genomics into healthcare systems.
Thank you. Bernard, why did you decide to focus on pharmacogenetics?
Well, that's also an interesting story, I would say. Initially, before jumping into pharmacogenomics, I I had a lot of interest around precision medicine overall. The reason why is, immunology, and most of the time the underlying causes of, let's say, primary immunodeficiencies is a genetic mutation. And during my career and training, and even residency training, it was a very few times where we were exposed to genetics, genomics, and most importantly, how to utilize that information. So that's why I pursue a PhD in immunogenomics in order to understand better, the underlying reasons of those diseases. And that's when where my passion around precision medicine started. Then I received my first patient with Dawes syndrome. And I think, as you can imagine, I was trying to understand not only how I can treat this patient, which I was already trained for, but how can I prevent that? And that's when, I connected the dots from the precision medicine piece and the power the pharmacogenomics may bring to those type of patients in order to avoid those type of critical clinical presentations. So, and that's been almost 14 years ago.
It's extraordinary listening to you both. I mean, it's the patient story that really drives a lot of the innovation, and it's great to hear that. Munir, I mean, you chaired this recently published report, which was looking at the implementation of pharmacogenetics in the National Health Service. What are the three main milestones you believe were instrumental in order to bring pharmacogenetics to a level where it can be seen as a key element of the National Health Service?
Thank you for that, Martin. So I think there are many different elements, obviously, and all of those need to come together. And we've highlighted all of those in our report. And just to give a quick background to the report before I answer the question, the report was undertaken in conjunction with the Royal College of Physicians, which represents 26 specialties, medical specialties, and also with the British Pharmacological Society. But we also had general practitioners involved in this. We had other colleges, anesthetists, psychiatrists, and pediatricians also involved in this. So we tried to be encompassing of all the different specialties which were important in this area. And it is, as I said earlier, it is important that pharmacogenomics is utilized in all different areas rather than in just certain areas. So that's why we had a very inclusive approach in developing this report, and the committee worked for a year online. We never met actually ever. It was during the pandemic. We worked online for 12 months in order to develop that report. And there are many recommendations, but the three main milestones, just coming back to your question, would be the first is funding, obviously, in order to implement this into any healthcare system, including National Health Service, there needs to be a pot of funding. And given that we feel the pharmacogenomics is an area where you can actually mainstream genomic medicine into a healthcare system, it's likely to be very widely used. And therefore, you need significant amount of funding to be able to make sure that, one can run a service which doesn't overwhelm the genomics labs, but also enables the people who are asking for the test to get the result in a short amount of time, but also the patients can get the results that they require. Clearly, the initial investment may seem a lot, but actually the cost effectiveness of that improving patient outcomes is going to be important in terms of overall cost benefit analysis as we move forward. So that's number one, is the funding. Number two is the infrastructure, actually, at the front end where the prescribers work. We've got the genomic technologies. I don't think that's a major problem, but actually it's getting the result into the patient's electronic health record, getting it to the individuals who are prescribing, and then providing them with the clinical decision support, which allows them to be able to make a decision as to what to do. So I think that's number two. I see that as, implementation within electronic health record systems and decision support systems. And the third is education and training. And this is, it's a rolling milestone, I think. It's not a milestone that, we'll be here, we can have a milestone which ends in 3 years. It's a rolling milestone. And the milestone needs to start right from undergraduate levels where you have to be able to change the curricula, bring into the curriculum of medical students, pharmacy students, nursing students, other clinical scientists, etc. But also then people during the medical training and other types of training, pharmacy training, nursing training, while they're getting the professional training, and also in time education required. The physician who's sitting there in front of the computer thinking what to do and then sees this pharmacogenetic test. It's good to have that relevant information available for that, just in time kind of information available for that individual, so that they can improve their knowledge and education. So I think it's training at the beginning in terms of educational qualifications, the professional qualifications one gets, but also continuing professional development as well. So it's a lifetime of learning in this area. I don't see pharmacogenomics as something which is going to be static. I see pharmacogenomics expanding. I see pharmacogenomics expanding in many different ways, with new drug targets, new sort of variants being identified, but also the use of polygenic risk scores as well as part of pharmacogenomics in the future. So it's going to be very dynamic, and people will need to keep up with the changes which occur so that it can be implemented into clinical practice in a seamless fashion.
Thank you. I mean, I love the statement, well, we've done the genomics, we can do the tests. I mean, it's remarkable change in 20 years to, from the first genome, to be able to hear a clinician really saying, yeah, well, that's sorted now. Now it's into the infrastructure and the education. Those are major milestones. Bernard, in terms of North America, do you see similar sort of processes and achievements?
Thank you, Martin. That was I have to say that I totally agree with Munir. I think it's amazing, and overall, indeed, those three main pillars of funding, infrastructure, and education/training are critical and instrumental for a successful deployment. Yes and no, to be honest. As you can imagine, the system is it differs a little bit, right? I would say, yeah, of course, the funding, it's there, and there are, several projects from different fronts going on. For instance, we have the All of Us project, US wide, bringing aiming to bring 1 million individuals in order to have, that data and help, modeling that information as well. Infrastructure, I think there are, from the technology perspective, we do have infrastructure. From the lab perspective, there are, top-notch laboratories in the US doing great exercise and efforts in terms of bringing pharmacogenomics. There's a lot of gaps that are about to be discussed, probably, in terms of integrating that infrastructure, interoperability within this infrastructure needs and pillar. Education and training is a pretty interesting piece because I think, when I was talking about pharmacogenomics probably 5 years ago, many of my colleagues thought that I was talking about Star Wars. I think not. I think we're getting into a momentum right now where it's not a strange topic anymore. I'm not saying there's full knowledge and that all the providers are comfortable utilizing it, but at least it's awareness about the possible benefits that will bring to the patient, and therefore they are eager to learning more about this and how to utilize it, clinically speaking. But there's the difference here are we need a clear, and this from the North American perspective. It's a different model in terms of not only who's going to be funding this, but who's going to be paying for this. So one key factor that may differ in terms of the US would be the coverage and reimbursement strategies. That's a big gap as well. And therefore, there are different stakeholders, a lot of different stakeholders playing here. So it's harmonization between the different stakeholders in order to provide a structured proposal for pharmacogenomic deployment, patient prescribing deployment. And last but not least, the regulatory landscape. I think even though it's clear, it's not yet there. So we need to have clear definitions, pathways, so that we can have as safe or as safe as possible approach when bringing this to ideally to the overall population. And last but not least, and I think this is also is something that might be aligned with the UK as well. It's how critical is diversity and inclusion. We know that we are multi-ethnical and multi-race countries, so it's critical to have the right representation of subpopulation in order to accurately deploy patient prescribing and pharmacogenomics.
Thank you very much. And I'll come back to you, Munir, a little bit about that. We know that a lot of the science that has led to guidelines on the use of common variants such as CYP2C19, 2C9, 2D6 have been done largely on white populations. Do you see more work being needed to actually implement into the multicultural population, either in the UK or further afield?
Definitely, that is really important, both in the UK but wider as well. I think 13% of the population in the UK is non-white, and it is important that we do not exacerbate health inequalities and race inequalities in the UK and more widely as well. So, you know, it struck home to me when we developed a warfarin algorithm, dosing algorithm, where we sort of developed it and tested it in a predominantly white population in a randomized control trial in the EU PAC trial between the UK and Sweden, where 97% of the population were white. But then when I was implementing this, I had to say, you can't use it in an Indian or African patient that comes to you. You just need to be able to use it in the white population. And so because of that, you know, I started doing some work in sub-Saharan Africa, you know, and working in Uganda and South Africa at the moment, and you know, trying to identify what particular pharmacogenomic predictors of warfarin there are. Also in other areas as well, for example, with dihydropyrimidine dehydrogenase, which is an enzyme which metabolizes 5-fluorouracil, or its oral analog, capecitabine. And if you have a variant in that particular gene, which reduces activity of DPD, then that can lead to 5-FU toxicity, bone marrow depression, you know, and other types of toxicities, which sometimes can be fatal. And the UK, during the pandemic in October 2020, introduced genetic testing for DPD throughout the NHS. That's in all 4 nations. That's England, Scotland, Wales, and Northern Ireland. You know, that's been implemented. We test for 4 variants at the moment. And we do 40,000 tests a year now. And that's in this of you know, in over 1 year while it's been implemented. We're doing 40,000 tests a year, which is great, fantastic in terms of implementation like that. But the 4 variants only test for individuals who are from a white background. They do not test for African variants. They don't test for Indian variants, etc. So that's something that we're working on at the moment. And you know, I've been working with the Race and Health Observatory, which is an NHS-funded body, and we are starting off an international consortium to see whether we can identify people of different ethnic groups, but also individuals of white ancestry, European ancestry, who have developed toxicity even when they were known to have either one of the 4 genotypes and the dose was reduced, or they were found to be wild type and they got toxicity, to identify new variants which are relevant for all populations, European ancestry, but also of African ancestry and other ancestry groups as well.
It's a very interesting approach to take people who have wild types of the variants you're testing for, but a reaction that's unanticipated with a particular drug, and start looking at those individuals in a way, rather than sort of waiting for these alleles to emerge from whole genome sequencing and other studies. Fascinating approach. That's very smart.
Yeah, hopefully what we're trying to do is to identify the outliers, you know, people who really have well-documented toxicity and identify particular issues in those individuals. And you know, I give you one example of a person who had one of the 4 variants, but then was given half the dose of 5-FU. But despite that, the patient unfortunately developed severe toxicity and died. And the family were keen to know what was going on, so we managed to get tissue samples and sequenced DPYD, and the patient had a rare variant, as you know, less than 0.01% of the population carry that variant. And so she was a compound heterozygote rather than heterozygote. And ideally, so you know, she should not have received 5-FU or much lower dose than 50% reduction.
I think this is a lesson that, you know, part of the implementation is going to be about using the information such as that to advance knowledge. That this is not a test that we just do, use the result, but it's part of an ongoing scientific piece of research, and it behoves all of us to be part of that exploratory program of using this information and then reporting on the impact. So, I mean, what do you think, Munir, are the next steps and processes that will need to be achieved in order to deploy pharmacogenetics through the NHS?
So, obviously, you know, we published a report. We are continually engaging with the NHS. The NHS has also funded a pilot project in the northwest of England. You know, and the pilot project is looking at some of those barriers that I talked about at the moment. And, you know, with that pilot, we'll learn what the potential barriers are, what the potential solutions are before it's rolled out. And this will be presented to the NHS. Clearly, you know, if you want to implement it, one of the things we said in the report was that it would be good that we implemented it throughout the whole of the UK, which is 68 million people. You know, and that's a big task, obviously. You know, and maybe it's overambitious. So it's good to do a pilot and see what the barriers are, etc., and what the best pathways are for implementation. Learn from that before you implement on a national scale. Clearly, so you know, we will hopefully identify the best pathways that allow us to be able to move forward. You know, part of the sort of ask will be the funding, as I mentioned before, in order to move this forward. But there are other things which are going on as well at the moment in the UK. For example, I know that one health board in Scotland has just funded its local area to be able to test for CYP2C19 in people who require clopidogrel for strokes. So you know, people are already starting to do that. NICE, which is National Institute of Clinical Excellence in the UK, is undertaking a guideline at the moment on CYP2C19 and clopidogrel in patients who've had strokes and TIAs. So it's coming through from different angles, you know, and eventually, I think you know, there will be a need for this, you know, as we suggested in our report, which is to basically undertake a reactive approach first, i.e., when a patient presents and is on a particular drug which requires a pharmacogenomic variant to be identified. But actually doing a panel approach allows you to be able to do the first test, but keep the other tests on the patient's electronic health record. So as the patient gets older, then you know you have the test data available already, so you don't have to keep on doing repeating the test. It makes it much easier for the clinician, it makes it much easier for the patient. And it is cost effective to do that, actually, because it doesn't cost that much more to do 40 variants than it does to cost to do one variant. So depending on what technology you use, obviously. So I think you know, and that means that we are moving from a reactive strategy to a preemptive strategy.
Yeah, interesting. And so, Bernard, you have 420 million patients to try and implement. Do you see a different approach required for North America compared, or do you see similarities of this reactive and then preemptive approach?
Yeah, I mean, definitely, it should be quite similar. In the understanding, the economical structure is a little bit different, but nevertheless, let me tie this concept to, you know, a little bit about around reimbursement and coverage, because then again, it's critical to define who will want to pay for this, That has been one of the biggest barriers in terms of implementing PGx is how can I make, you know, my insurer, either private or Medicare, Medicaid, cover my testing, right? So that's been one of the many problems here. It's I think what Munir mentioned. It makes a lot of sense from cost effectiveness, total cost of care savings, being more efficient, and most important quality of care, and that is panel approach on an evidence-based fashion, utilizing specific guidelines and recommendations with evidence behind them. I think there are enough information to actually bring panels instead of single genes. Nevertheless, that's one of the first discrepancies that I'll be talking about between the clinical/scientific evidence against the reimbursement coverage determination evidence. So most of the NCDs, LCDs that are in the U.S. national or local coverage determinations, for instance, most of them are based on specific. Most of them, number one, are restricted, and secondly, they're based on specific genes, and that's a big problem. That's a big problem, right? So because it's not affordable. You cannot just test. Let's say that you have a massive deployment and you test 200 million people for one gene, and then you need to go to CYP2C19 because now the evidence is there. You will need to do that again. So just as simple as that, but it's not going to be reimbursed. So therefore, it's one of the limitations, one of the barriers that we have been facing here. That is the harmonization, the criteria harmonization between the clinical and scientific working groups with the payer overall scope.
Yeah, very interesting. And so, I mean, just going back to that bit about, well, we're not sure what the alleles are yet. It's even more than a panel, perhaps, for not maybe 200 million people, but we're talking about being much more comprehensive in our testing so that we do have those unusual alleles detected for other populations.
Yeah,
I mean,
that's
go on.
No, I just don't think that's critical, to be honest. Have the right coverage. There are many genes that we need to be certain that the panel or the laboratory testing that we're utilizing in this case, there are several options for North America, has or it brings that coverage, CYP2C6 and those specific alleles that are may differ from a Black patient to a Southeast Asian patient or Hispanic patient. That can change the phenotype and therefore the recommendation. Yeah. So the standardization, again, it's something that it's a massive need.
So I think it's important to start somewhere, isn't it?
Yeah.
And although the technologies are there, from single SNP testing to panel testing to sequencing to exome sequencing to whole genome sequencing, long read sequencing, etc. So you've got to start somewhere, and what's available at the moment and what's going to be cost effective at the moment. I guess what we're doing is moving in stages. Relatively rapidly, actually, to having whole genome sequencing in everybody. When whole genome sequencing becomes $100, then everybody will have it, and so on. Yeah. And it's then a bioinformatic challenge to be able to identify those relevant variants and provide them to the clinician. And as you know, and I know you want to talk about Genomics England in a minute, Genomics England was able to sequence 100,000 individuals, and they're looking at the pharmacogenomic information in there and how they can relay that back to the healthcare system. So that when everybody has a whole genome sequence, then we don't want that data on pharmacogenomics to be lost. We want that to be extracted in a way that enables the clinician to be able to then undertake the prescribing as well.
Yeah, it's so important to have that information flowing with the patient wherever they are and whenever it's needed. Totally. So in the UK, David Cameron, previous Prime Minister, set up Genome England, which was a company owned by the UK, but or owned by the National Health Service. So is and do you see that sort of approach being necessary, a public-private partnership? And maybe you could talk a little bit about that.
Yeah, so Genomics England was set up, as you know, at the time of the London Olympics. Or that's when it was announced, and it was set up after that with the aim of really transforming and providing genomics to the population. through whole genome sequencing, and it focused on whole genome sequencing of patients with rare diseases and whole genome sequencing of people with cancer. And they undertook for people with cancer, they undertook sequencing of the somatic genome as well as the germline genome. And that data was then made available to the clinicians at the front line in the NHS. And that was transforming and providing genomics right to the front line. And so that's been extremely successful, for Genomics England in terms of what they're doing. Now they are working with the NHS, Genomics England, and providing the services behind some of the whole genome sequencing, which is going on as part of clinical practice in rare diseases, in certain cancers, and in children with cancers as well. For example, the UK has said that they will sequence all children with cancers and so on, and provide that data to clinicians. And Genomics England is a company which is wholly owned by the Department of Health and Social Care in the UK and provides funding to the Genomics England, but obviously Genomics England works with lots of companies as well in order to do the genomic sequencing and things like that. So, but if you look at pharmacogenomics implementation, what you want is solution, which is an end-to-end solution. So if you look at the laboratory where you're going to do the sequencing or genotyping, then obviously you're going to be working with companies, with the platforms in order to undertake the genomic genotyping. Then if you go downstream to how you're going to then provide that data to the clinician, then you'll be working with companies that do the clinical decision support and so on. So I think there's going to be a lot of different companies along the way. So it might not be a single PP, public-private partnership, but it will be a partnership between the NHS and many different companies in order to provide that end-to-end solution, which allows the final end product that allows the clinician to prescribe the right drug and the patient to get the right outcome.
Thank you. Bernard, in terms of North America, do you see a public partnership, a public-private partnership emerging, or do you think this will be completely private or mixes? How do you see it actually playing out?
I think that's the only feasible way, to be honest. I think if we want to have, a population-based deployment here and a population-based scale, that is the only feasible way, at least from my perspective, where both we need to, bring our muscles together in order to make it happen. And I think to some extent it's already happening. I think there are, private organizations doing fantastic things around population genomics, for instance, great centers, educational / academic centers, healthcare systems aiming to bring this their specific subpopulation, and different companies bringing the rest of the IT technological factors, as Munir mentioned, trying to face these barriers. Nevertheless, the big problem right now, or the biggest problem, is actually, communication between different stakeholders here in order to standardize the approach. And then, two key elements that play a massive role, technically speaking, that is, portability and interoperability. And that's another thing that should be also addressed accordingly.
Yeah, absolutely. And I mean, Munir, you just touched on clinical decision support. I mean, what is the role of clinical decision support in this initiative within the United Kingdom?
Well, it's vital because most people who are prescribing, doctors, nurses, pharmacists, won't have the detailed knowledge of pharmacogenomics. So one needs a clinical decision support system that interprets the variants and then links to guidelines, which says this is the next thing to do because that patient has that particular variant. I think that's important. If doctor, nurse, pharmacist had to do that manually, he would then have to look up what that variant means. He would then have to, he or she would have to then determine what to do as a result of that variant. That would take them at least half an hour for each variant. And if you consider that in the UK, I think a general practitioner has about 10 minutes to see a patient, probably less than 10 minutes. As a hospital doctor, I usually have 15 minutes to see a follow-up and 30 minutes to see a new patient, and where you look at all those different things, then you can't really fit that in if you're going to do this manually. So you need an automated process which makes it as easy as possible for the clinician to make the right decision. And the IT systems are going to be critical, and the clinical decision support system, which is user-friendly, which has been user-tested, etc., and which is able to be interoperable with the relevant systems. In the UK, we have two main systems within primary care, but actually in secondary care, we have many different systems that are there. So we need to make sure the interoperability is there for whatever system's there, so that it can be fitted in, if you like, inverted commas, seamlessly into that particular healthcare setting.
Yes, I'm looking forward to a seamless integration into an electronic medical record. But I think we need to aspire to it, absolutely. And I think, to your point, I remember that there's a paper by Yusuf looking at the number of genes and drugs from using quite a small limited panel, and eventually coming down to, if you implemented this preemptively, it was about, for a normal family physician or general practitioner, about 12 patients a week where you might be using this information. So yes, you needed something that could help you deal with this a lot faster than a half hour per variant. Yes.
And I think 12 patients is probably an underestimate, actually.
Yeah. Bernard, in your experience, clinical decision support, I know it's a loaded question and I have to put it out there. You've got a name behind you that indicates a slight bias in favor of clinical decision support.
Yeah, that's correct. But I can go beyond that, to be honest. I can use my information hat. I think, the wow effect of pharmacogenomics was, also a pitfall. 15 years ago, we all thought, the PGx will just be as disruptive in a way that, no more adverse drug reactions, efficiency or efficacy, treatment efficacy would be 100%. And, this was a game changer. And the reality, it's good, but it's just another piece of the puzzle. So that's also from the clinical perspective. We are not intending to change the clinical workflow. In fact, we are, as Munir mentioned, we're trying to streamline it in a seamless fashion. And that's what a CTS can do. And bringing pharmacogenetics is just another piece of the puzzle that of the data sets or the factors that matters when prescribing. So I think, it's at least from my perspective, is the most suitable way on how we can efficiently deliver PGx in a way that I don't need to be an expert to understand and utilize it, that it can be, part of make my decision an informed decision, such as with the other other factors, kidney, liver, drug condition interactions that we need to take into account when prescribing.
Yes. Munir, in the UK, is that also recognized that this is part of a puzzle, that the pharmacogenetics on its own is one part of the solution, but we need to integrate the other factors into the process?
So yeah, it's obviously, from my perspective, yes, it is. It should be recognized. Whether it is recognized or not, I'm not sure. But I think pharmacogenomics is just one part of the puzzle. There are many other things which determine variability to drug response. Your age, your other diseases, your renal function, your hepatic function, what other drugs you're taking, etc. And all those need to be incorporated in kind of multimodal algorithms, including the genomics, so that we make the right decision. Because we at the moment, most clinicians, or particularly in primary care, will have specific prescribing systems which provide you with knowledge on drug-drug interactions, right? And obviously drug-drug interactions are just one aspect of it. There are drug-gene interactions, then there are drug-drug-gene interactions, then there are drug-drug-gene-gene interactions, etc. So it becomes more and more complicated, and we need that built in to all systems, so that allows us to be able to have that support and the right decision at the end of it. And to look at pharmacogenomics in isolation, in some cases might be the right thing to do. For example, with HLA, with the dress, that Bernard mentioned. Abacavir is such a strong predictor of abacavir hypersensitivity, etc. But with many others, you need a multimodal algorithm, and we developed that with warfarin. We had 3 alleles, we had age, body mass index, drug-drug interactions, and other algorithms have had other things in there as well. So we need to be able to develop those kind of algorithms within our decision support system, which allows us to be able to then decide on what is the best dose for the patient, what's the best drug for the patient, and what is the right time, hopefully, as well for the patient to get that particular drug or drug combination.
This is a complex process, and we've touched on the prescribers and who they are. You talked about physicians and pharmacists. This is a big group. How do we bring those together? Do you see any challenges in doing that and sort of harmonizing approaches from specialists, primary care, hospital pharmacists, community pharmacists, so that they're all doing similar things using similar approaches?
Yeah, so obviously different people will have different roles throughout that kind of spectrum of people that you've just described. And if you look at super specialists, they will be dealing with particular disease areas, and they probably won't need that much decision support because they'll be dealing with a very small number of drugs, working in their particular area. But then you go to a general practitioner who's looking at a multiple number of diseases, probably prescribes a much larger number of drugs compared to the average specialist. Then they need a different kind of information and availability of that information. I don't see that there is any kind of challenge in terms of interpersonal or interprofessional challenges. I just see that what we need are the relevant support systems so that people can undertake their particular clinical practice and make sure that the clinical pathway is not too disrupted. Clearly, it will be disrupted to some extent, but I think it is important that we fit in whatever we want in order to improve the prescribing, in order to improve the dosing in a seamless way into the existing clinical pathway. And that's important. And I'll give you an example of that. When we implemented the warfarin pharmacogenomic testing in UK peripheral clinics, what we did was to have a point of care test, which we trained the nurses and pharmacists to be able to use. They were able to get the results in 45 minutes. Doing the mass swab, they put it into the point of care test, and then they got a result in 45 minutes. They were able to put the result into a web-based algorithm, which told them what the dose should be for the first 3 days for warfarin. And we were able to show that using that kind of approach as an implementation study, we had already done the randomized control trial. This was an implementation study. They were able to get the same result as the randomized control trial, a 7% difference between individuals who were genotyped versus individuals who were not genotyped. But as part of an implementation study, we also did questionnaires. And they all liked the genotyping and they all thought it was a good thing. But the nurses complained of the fact that it lengthened the clinic because the patient had to wait for 45 minutes for the test result. Right, so the patient had the test genomic genotyping done, then had to wait 45 minutes and had to come back. So it lengthened the time the patient was in the clinic. They had to go and get some coffee or something like that, and then they came back to the to the clinic. So overall, lengthened the time of the clinic. So they didn't like that. So we need to think about how we can actually make sure the clinical pathway is rationalized, optimized, so that both the patients and the clinicians who are using it like using it rather than thinking that it is making their life worse.
Yes, unintended consequences. It's always interesting. Bernard, do you see differences in North America, the interprofessional relationships, the people using the pharmacogenetics?
I have to say sorry about that. I have to say from the professional/provider perspective, I totally agree with Munir. It's a pretty similar approach, and I don't see a big problem there. I think even nowadays, even more, the close relationship between the pharmacist and the physician, it's improving by utilizing this as the catalyzer of that relationship. I think the problem goes beyond that. I think the problem is more about the fragmented system that we currently have and the complexity of communicating A to B. And the bricks and mortar sometimes doesn't allow that dynamic interaction as we have been describing. So I think it's more, it has become, or it's becoming a technological issue that we need to address accordingly. It's a thing that probably, the new trends, quote unquote, new of, start utilizing cloud with us, safe, obviously with all the safe credentials that we need, a cloud approach in order to try to bring that information together and assess, compile, utilize, and organize in order that the clinician and the different clinical workflows can utilize this is the next step. So long story short, I think the problem is beyond the providers or the healthcare providers specifically, and now it's more about the system per se and the interoperability as we briefly discussed before.
Yes. Yeah. Thank you. And I do have some questions coming in from the audience. One is, how can we convince physicians to recommend pharmacogenetic testing, as many of them mentioned to medical associations that do not consider pharmacogenetics? So I think getting, maybe the different specialty organizations around the world to get more interested and involved in pharmacogenetics. So maybe, Munir, have you experience with that? Was it easy to get the Royal College of Physicians and all the specialties involved in that report?
So it was not a particular issue to get the Royal College of Physicians involved, but we, so the people who then sat on the committee were those who were already knowledgeable of pharmacogenomics and were convinced that pharmacogenomics is an important aspect to look at and implement. But clearly, there are a lot of other people who still need to be convinced that pharmacogenomics is important to implement. And I guess, what we work on still is the kind of evidence-based medicine hierarchy, where you have randomized controlled trials at the top of the evidence hierarchy, while with case reports, ideas, opinions at the bottom of that triangle. And I think that we need to move away from that kind of hierarchy, and we need to treat all evidence in a very intelligent manner and look at the sort of quality of evidence and whether the evidence makes sense in order to be able to change practice. And using real world data, and we've got more real world data now than we've ever had. And so that big data approach is using that intelligent way to be able to understand where things are working, where things are not working, is going to be important in order to move forward. So I do think that we do need to move away from the kind of evidence-based hierarchy. Clearly, many of the guideline developers require randomized controlled trials when they change the evidence base and so on. But can we really do a randomized controlled trials for every gene drug variant? That's going to cost billions of dollars to be able to do that. There's no way one can afford to do that. So we need to be able to use data from all sorts of areas to be able to do that. And when you look at polygenic risk scores, which may be coming in for particular drugs, they're coming in for disease stratification, etc. already. But when you start looking at those, then there's no way you can undertake randomized controlled trials for that, for all of that. But so we will be using data from all sorts of areas, real world data, but it's important that we make sure that we have the relevant systems in place to monitor the impact of that. And so, if we have the good IT infrastructure, we can actually develop a learning health system whereby you can introduce particular genomic variation into your clinical practice, and then you can follow the patients through to see whether it's having the relevant effect you intended, identify any unintended consequences, and if you do identify any unintended consequences, identify what the problem is, then go back to your initial panel or genomic test and modify that so that you can actually get rid of the unintended consequences. And so what I'm suggesting for the future is that we will have all sorts of data, including randomized control trials. We will need to use real world data in an intelligent way. And when we implement something, we'll need to follow that through using the infrastructure that we have, and we can develop even further so that we develop learning health systems, which allows us to be able to modify and improve, continuously optimize what we're doing in terms of the kind of genomic testing, but also in terms of prescribing as well.
Yeah, thank you. Bernard?
Yeah, I think that's brilliant. I couldn't agree more, to be honest. I think nowadays we have the tools. The information is out there. The data is out there. I think the next obvious steps would be, number one, as we have been discussing, continue fostering collaboration beyond borders, continue fostering collaboration. Number two, obviously structuring that data and organizing that data so that we can bring the artificial intelligence piece into this. I think that's a key factor for not only the randomized controlled trials, you can prove a concept by modeling data, but also there's many things that you can bring clinically speaking into point of care by utilizing AI. Therefore, the ones that are creating data, we need to be aware, number one, that data should be transparent. I think that's critical, transparency about the algorithms. We are, those black boxes, proprietary information. It's okay if that's the business model, but you need to be able to clearly define how does your system work. You need to be clear. You need to be able to transparently define how you're taking point A to point B in order to make set or whatever. And obviously, take into account the bias effect. Remember that AI is it's taught by humans. So it needs to be critical that we are teaching the machine how to learn, where to learn, and as unbiased as possible. Because if not, there's a big risk, a big risk of fostering disparities instead of tackling those. But I think it's fascinating times, and this is a train that has departed, and I'm thrilled, with what I've heard from the NHS and what Munir is sharing. I think it's amazing, and it's something where we all can also learn a lot.
Thank you. And a question that's just come in from Adriana Ketchik, talking about who are the best candidates for pharmacogenetic testing. And I think that's a really good question, and I think I'm kicking myself. I didn't actually put that into the list of questions, but Munir, I mean, you mentioned a couple of examples there, but who do you think we should be doing pharmacogenetic testing on?
Okay, that's a great question. Thank you very much for that question. So if we just take a step back, it's important to realize, and the data which is coming from all parts of the world, is that almost all of us carry some kind of pharmacogenomic variation. So 99% of us carry at least one pharmacogenomic variant. A lot of people carry at least 4 pharmacogenomic variants. So pharmacogenomic variation is common in the population. When you look at the veterans study, which was in about 1 million individuals, they found that 37% of those individuals were on genes where there was good evidence that pharmacogenomic variation had an impact on outcomes, 37%. But that's a sort of wide age group in the veterans study. When you look at UK data, and this was work undertaken using CPRD, which is Clinical Practice Research Data Link, what that showed was that by the time you get to 70, at least 90% of the people over age of 70 are on at least one drug which is affected by pharmacogenomic variation. So from that, then one can decide what the eligibility criteria are. And obviously the eligibility criteria for pharmacogenomic testing may vary according to different healthcare systems and according to different drugs as well. And in order to help with that, we have developed some eligibility criteria with the NHS for particular drug gene pairs. So you may come in with one particular drug you require, and you fit in the eligibility criteria, then you can have the pharmacogenomic panel test. Hopefully that's how it may work in the future. But your genetics is then stored in electronic health record for the future drugs that you may be exposed to. And that those eligibility criteria can vary according to which different drugs you're looking at. So, for example, drugs such as clopidogrel, I think if you're putting somebody on clopidogrel, for let's say for stroke prevention, then I think everybody should have 2C19 testing when you go into clopidogrel. If you're putting somebody on warfarin, I think you should have the testing to see what dose you require. If you're putting somebody on abacavir, you need HLA-B5701 before you put patients on abacavir. So I think that is important. If patients are already in the 70s, they'll be on multiple drugs, then maybe those are the particular groups that require pharmacogenomic testing as well. And we're testing that at the moment to see whether, when you recruit people who have got multimorbidity, more than 2 diseases, and are on more than 5 drugs, what proportion of those really do require pharmacogenomic testing? So we're actually undertaking that study at the moment, and hopefully the results for that will be available. But as you also know, that we undertook the UPGx trial, which was looking at people coming into hospital and who are being prescribed particular drugs, pharmacogenomic drugs with pharmacogenomic variation, which were mentioned in the Dutch Pharmacogenetics Working Group guidelines. When they were then enrolled onto the study, they got a panel approach, whereby they got the result for the particular drug that they were started on, but then the rest of the results were made available to the clinicians for future pharmacogenomic testing. And that's the way I see it going, is that you do it on the basis of the initial drug that the patient is going to be put on. If there's evidence that drug has pharmacogenomic variation, then you should probably consider pharmacogenomic testing, and then hopefully have more pharmacogenomics data available in the patient electronic healthcare record for future use.
Yeah, thank you. And I'm just going to squeeze a quick question into you as well, Munir. It's coming about the education, and they talk about, yes, we need education for the professionals. Ask, what about the public? Does the public need education on what's happening in the UK, particularly for that?
Yeah, so we were very keen. We had a public member on our panel as well, on our committee, to get a public perspective on this, and we have a lay summary associated with the report. And we feel that public education is really important as part of this. And in terms of future design of the pathways of the service for pharmacogenomics design in the UK, we said that patients should be involved in that pharmacogenomic design of the service, because they know exactly what it feels like to be a patient and where the barriers are, and so they can help in terms of make sure that service is as equitable as possible, as efficient as possible, and hopefully acceptable as well to the public as well. In terms of the general public, it's important for them to be aware that there is variation in drugs and the way people respond to drugs. I think most people know that. Most people have heard of personalized medicine, but what they probably don't understand is that personalized medicine is not a magic bullet. It will never, there's no test that we will ever have that will guarantee that you are going to respond exactly, 100% to that drug. So, but what we are doing with pharmacogenomics and other types of precision medicine is going from where we are now, which is really very little predictability, usually less than 10% predictability, to more than 50 or 60% predictability, which is really important, because we need to be able to move away from the kind of trial and error approach to something which is more predictable.
Absolutely. Bernard, comments about public education in the last minute or two?
Yeah, Bumpy, may I just say something before that briefly? I just want to say that, in a nutshell, being bold and disruptive here, the reactive approach that we're having in healthcare is not working. The numbers, show that. So, predictive approach is the one that we should be taking. So, having said that, if you need to prove a concept, the low hanging fruit, maybe, mental health, can be cardiovascular conditions, can be oncology, and a specific, HLA or specific, highly penetrant gene drug interactions, as Munir mentioned. So, but ideally, if you want to be disruptive, everyone can benefit from testing. In terms of education, that's right. People need once the patient knows about this and owns it, you power adherence and trust, and that's a big problem. On any specialty, the patient trusting the physician, trusting the treatment, and waiting for that treatment to actually to have the expected outcome. So by bringing this extra tool, you also are fostering adherence, which is a key factor for success as well.
Thank you. This has been a phenomenal conversation. Two great experts, Munir. I really appreciate you spending part of your hot evening in the United Kingdom with us, talking and obviously you're so enthusiastic about pharmacogenetics, and that really comes across. And your expertise and your knowledge has been really helpful in this conversation. Bernard, thank you for providing the North American translation and interpretation. It's been an amazing session. We've got a lot of questions, which I'm sorry we couldn't get round to, but indicating lots of involvement from the audience. So thank you very much for all of those who've attended and your questions, and we'll do our best to respond to those over time. Thank you again.
Thank you very much.
Thank you.
Bye-bye.